Hi everyone, Jeffrey here. I'm going to be talking to you guys today about DNA replication, and I'm going to try to make it, you're going to have a basic understanding of DNA replication, and I'm going to talk about the nuances about it today. So to get started, DNA replication is basically where you take a double-stranded piece of DNA and you make two double-stranded pieces of DNA from it. And how you do that is through semi-conservative replication. What that entails is that you're taking an old strand of DNA from one of the DNA strands in that double-stranded DNA helix of the DNA that you're replicating, putting into one new strand of DNA, and taking the other strand and doing the exact same thing. What that means is the resulting product is that you have two DNA strands, but they both have DNA from the original parent DNA, if you will. What we're going to talk about now is the basic unit of DNA synthesis, which is the nucleotide. It consists of your phosphate group, a ribose sugar, and a base. That base, as you guys know, ACTG. And I labeled out the 1', 2' through 5' numbers, so it allows you to have a little bit of an understanding of when we say DNA synthesis is only 5' to 3', what does that mean? We're looking at what's on the 5' and what's on the 3'. And what is on the 5'? It's a phosphate group, and the thing that's on the 3' is that OH group. Okay, and we have three phosphate groups. Two of them get cleaved off when you're adding a nucleotide to a DNA sequence. That provides the energy actually for it to create that phosphodiester bond. Many of you guys have probably heard of a phosphodiester backbone, and what that means is you have a phosphate group, it's forming an ester right here, it's also forming another ester right here. That makes a phosphodiester bond. Okay, and synthesis is always 5' to 3'. If there's nothing else that you learn from this video today, it's that synthesis is always 5' to 3'. And in order to synthesize 5' to 3', we need to read the DNA strand from 3' to 5'. So in this template strand, you can see that there's the 3' end, here's the 5' end. You can see that the OH group is right here, the phosphate group is right here, and you're reading it 3' to 5', and you can see that the synthesis here of the primer strand is 3' to 5'. Okay? And you can see that the pyrophosphate is cleaved off. That provides the energy to form that phosphodiester bond. Alright, so now let's talk about DNA replication in prokaryotes and eukaryotes. So what I want to really emphasize today is really the differences, because you guys have a general understanding of how DNA replication is formed and the general functions that all of these enzymes serve. Alright, so let's talk about bacteria real quick. There's two different types of replication. There's going to be theta and there's going to be a rolling circle. And I'm going to use a different color now because black on black is a little hard to read. But theta and rolling circle, learn on your own. We don't have enough time to go over that, but it's fairly simple. You should be able to do it learn it in 5-10 minutes. Let's talk about the enzymes. Initiator, or DNA. What is an initiator protein? Essentially, initiator proteins help you initiate the start of DNA replication. So initiator proteins first bind to that DNA, and then adjacent, the DNA adjacent to that, remember that the bases, when they, like if I have A and I bind to T, for example, I have two hydrogen bonds, and essentially what initiator proteins do is it kind of denatures those hydrogen bonds so that it opens up this space right here adjacent to the initiator protein and allows for DNA helicase to go on and attach itself. And we all know that DNA helicase is the thing that unzips all of your hydrogen bonds, and as we affectionately say, it unzips your genes, right? So you have your DNA helicase, that's also called your DNA B, it is delivered via the helicase inhibitor. So what's not shown here is that DNA-C, or the helicase inhibitor, is the thing that's actually recruited and delivering an inactive DNA helicase. And once it delivers that helicase, that DNA helicase is going to be viable, and it's going to work. Okay. RNA primase, we all know that DNA polymerase 3, as great as it is, it cannot de novo synthesize. What de novo synthesis means is that you need an open OH group to synthesize DNA. So in order to add on the different nucleotides, you would need a free-floating OH. And if you're just synthesizing, if you're just unzipping DNA, so for example, if this yellow was DNA, DNA polymerase 3 can't just start synthesizing DNA from a template without a primer. So what RNA primase does is that it lays down a groundwork and allows for the DNA polymerase 3 to do its job. Okay? Great. So you have your DNA polymerase 1, which removes your RNA primer. So that's the primer from the primase. And then DNA polymerase 3 synthesizes your leading and lagging strands. So this is the big thing that we're talking about. And both of these, DNA polymerase 1 and DNA polymerase 3, they have 5 to 3 activity. So DNA polymerase 3 does synthesis. DNA polymerase 1 excises the primers, the RNA primers. So it excises the RNA, but it also synthesizes DNA. Okay? It just does it more slowly than DNA polymerase 3, which is why DNA polymerase 3 takes care of the leading and lagging strands. And then you have your single-stranded binding proteins. Why you have that is that when you have your helicase and it's chugging along, the DNA can actually bind with itself. So if this nucleotide was, or if that base was a C, it can bind to this base if that was a G. So then you'll form these hairpin structures, and when your DNA polymerase comes chugging through, it'll completely just skip over. So this, you can see that this area is just completely skipped over. It's not replicated. That's not something that we want. We need to replicate all of our DNA, right? So your single-stranded binding proteins come in. It allows for none of that hairpin structure to form. And then you have your DNA ligase that comes in with your lagging strands. So DNA ligase helps with that. Okay? And then you have your clamp protein. So what a clamp protein is that you have, let me erase this real quick. So you have a DNA polymerase, but it actually has a pretty low affinity. It can fall off pretty easily. And what you want to do is, obviously, you want it to synthesize your DNA. So you have a clamp, and it'll essentially just clamp down your DNA polymerase to the DNA strand that you're trying to synthesize, right? And then you have your DNA gyrase. It's a topoisomerase, and topoisomerases relieve the torsion, and that's going to be further down. So if I'm synthesizing a DNA strand and my helicase is right here, my topoisomerase is going to be downstream, and let's say like this is my replication bubble. Okay. For eukaryotes, it's going to be linear replication. The enzymes are very much similar. The only things I'm going to talk about are the things that are different from eukaryotes compared to prokaryotes. And what's different is that it has DNA polymerase alpha, which is like the RNA polymerase from bacteria. Okay? It synthesizes RNA primer, has the exact same function. DNA polymerase sigma is like the DNA polymerase 1 and DNA polymerase 3 from prokaryotes combined. So it can not only remove RNA primer, but it can also synthesize your leading and lagging strands. Your topoisomerase, your type 1 breaks 1 DNA strand, and your type 2 breaks 2 DNA strands. So essentially, to relieve your torsional strain on your DNA, you essentially break the DNA strand, uncoil your DNA, and then attach it together again. There are very popular, very short videos on topoisomerase. It'll explain the difference between type 1 and type 2 quite well. Google it. And the animations are going to be a lot simpler for you to understand than me talking about it. So now moving on to replication fork, I'm just going to talk about this very briefly and very broadly, okay? So you have your helicase, you have your helicase. It's going to unzip your DNA this way. It breaks all the hydrogen bonds.
I found this on the web.
Okay, thank you, Siri. And then you have your SSBPs, which are your single-stranded binding proteins, keeping your DNA from forming those hairpin loops. You have your RNA primase synthesizing those RNA primers, which that one's drawn in yellow. Sorry about that. And then your DNA polymerase 3 synthesizing these DNA strands. Okay, and the continuous strand, which is the top one in this instance, because and it's also called the leading strand. It's also, so it's continuous because you have one primer and then it'll go with the flow the entire time, because the template strand is 3' to 5'. So because the template strand is 5' to 3' on top, you're going to have synthesis from 5' to 3' this way, and that's going to lead to continuous synthesis. You guys already know that. And then for lagging strand, it's going to be discontinuous, and the reason why it's discontinuous is because as your helicase keeps going down, you're going to keep on having to synthesize all of these different primers so that you can actually go from 5' to 3'. Remember, DNA synthesis is always 5' to 3'. And as the bottom DNA template is 5' to 3' in this direction, that means your synthesis has to be in the opposite direction, right? So as your synthesis is in the opposite direction, that's why you have your lagging strand, because it has to go in this direction, 5' to 3', and it needs a primer every time. You can see that it's very choppy, and that it's going to lag along because it's going to take more time to put down that primer, and then as compared to this, where you only need one primer. Okay, hopefully that makes sense. These things highlighted in blue are Okazaki fragments. Basically, they're just part of the lagging strand. So right here, we're going to zoom in a little bit, and we're going to talk about what happens with the lagging strand here. So this is going to be the lagging strand. It's the exact same thing as on top. You have your 5' and 3' on the bottom here, and this is your template, right? And on top is you're going to synthesize your DNA. So you have your primers, and then you have the DNA that's synthesized. So now you need to remove the primers because this is RNA and the blue is DNA, and we want all blue in this instance. So what happens is that we need something, and you guys already know this, we need DNA polymerase 1 to come in and synthesize DNA from RNA. So it has that 5' to 3' exonuclease activity, as we mentioned before. So it'll excise all of this RNA and make it all DNA. So it'll do the same thing right here. It'll do the same thing right here. But there's going to be a problem. And that problem is the DNA polymerase 1 is going to fall off once it reaches the end of every single DNA sequence, right? So what happens there means that you're going to have a missing phosphodiester bond. Okay, so you're missing your phosphodiester bonds, and you're going to need something to help make that discontinuous DNA continuous, because you can't use it unless you have all these kinks filled in. So what happens is you have this thing called DNA ligase, which comes in and it will repair that kink. It will restore that phosphodiester bond, making your DNA continuous. And it does this through a process called phosphorylation. So it uses ATP. It uses the energy from ATP to zap that bond into place, to form that phosphodiester bond that it's missing. Okay? And this is going to be a replication bubble. I'm just going to talk about, your topoisomerase and your gyrase are going to be here. Your helicase is going to be here. And this is just a rough representation. And I think what I want to talk about here is just some key points. Okay, so this is the template strand. These two are the template strands right here. And you have this going from 5' to 3' this way, and 3' to 5' this way. And remember that DNA synthesis is always 5' to 3'. So if this is 5' right here, then this is going to be 3' here, 5' here, 5' here, 3' here, right? It's going to be 5' to 3' synthesis. Now we're going to have, 1 primer here, and this is going to be a leading strand, because 5' to 3' continuous synthesis, we don't need another primer after we've already made our primer here. But as we open up in this direction over here, this is still 5' to 3' synthesis. It's just that now it's lagging because we need to synthesize new primers down the line every single time, right? So that's why we have a lagging strand here and a leading strand here. And that same concept applies to the bottom strand here as well, because as DNA helicase continues to break those hydrogen bonds, we're going to have to continually synthesize new primers before we can synthesize that DNA. So as this bottom strand is 3' to 5', you're going to have this being 5' to 3'. So that's 5' to 3' synthesis. But once we reach this middle right here, this origin of replication right here, you can see that it's going to be still 5' to 3' synthesis, but now all of a sudden it's going to be continuous, and it's going to have only one RNA primer before you can go through that DNA. Okay, so now the lagging strand is going to be on this side, and the leading strand is going to be on this side. I hope that clears some things up. I know this is really messy. I know I rushed through a lot of huge concepts here, but I just wanted to give you guys a broad overview as well with these nuances right here. That the nuances were extremely important and something that I wanted to touch on. All right. If you have any questions, leave them in the comments below and I'll get to them and I'll try to answer them. All right. Thanks.